This is Immuno Africa, a podcast dedicated to spotlighting African immunology research.
Sometimes what opens the door is the confidence for you to even, because people are like, how dare you? There must be something about you that made you knock on this door, because how dare you? If you've got one leg in, use that leg in to push your entire body in.
Welcome to the Immunology in Africa podcast. I am your host, Welt Okete. If it's your first time here, kindly support our work by subscribing to this podcast and turning on notifications to keep up with new episodes. We're currently on Spotify, Apple Podcast, YouTube, and other streaming platforms. Dr. Ogbe is an Associate Scientific Director at Moderna, previously a postdoctoral immunologist at the University of Oxford. Our work contributed to current understanding of HIV and COVID-19 immunology. However, contrary to this career trajectory, Dr. Ogbe didn't always think she could be a research scientist. After her undergraduate program, she was eager to pursue an MBA, not only because she lacked access to relevant opportunities, but also because she doubted the quality of the training she had received. How she got here and what she's been up to make up the bulk of our chat. But first of all, let's meet her.
So just to say, before I start that, my views, whatever I share here, represents my own views, not the views of my company, not the views of any organization. So these are just my personal views. So I am a Nigerian, grew up in Nigeria, lived in Nigeria up until the age of 24 or so. Had all of my primary, secondary education in Nigeria, and then moved to Ahmadu Bello University for my university education. I did a bachelor's there in biological sciences, I think it was called then, and specialized in just general biology. And then and I guess this is why I like to support people that read the natural sciences or study the natural sciences at university level in Nigeria. Because when I did it, I think the prospect for me then from a lot of people was, oh, so what are you going to do after this? You're going to be a teacher or a banker, and there's absolutely nothing wrong with any of those professions. But I'm just trying to say that, there are more options for people. You don't have to be limited by the choices that are available in Nigeria. And while I've got a passion for teaching, mentoring, and lecturing, and all of that sort of stuff, I think I wanted to have broader options. So I did go into banking for a few years, and then got an opportunity to study abroad. I was privileged to have that opportunity. I do appreciate that that's not an opportunity that everyone has, but I was privileged that my parents were able to send me to the UK to study. And I remember thinking, okay, I'm going to study an MBA and come back so that I can carry on with my profession in the bank. And my dad said, I'm not going to fund your studies if you're doing an MBA. And at that time, that was like the most devastating news I could have ever received. So I thought, okay, what else do I want to do? And for me, it was cancer research. That was the buzz at the time. I used to sort of go on the Cancer Research UK homepage and read it up from Nigeria, and it just sounded so fantastic. And I remember dreaming like, oh, if I could get to work here, it just sounds so exciting. And so I decided to apply for a master's program in cancer research, and I chose Brunel University for my master's program because the course sounded interesting. The content of the course, the location was really good, and also I had some sort of my accommodation covered by some of my relatives for a while, so I got in. And when I speak about the education in Nigeria, I think that was the first thing that hit me when I had an assessment to do, my very first assessment. And the way I was trained in my university in Nigeria, we called it cram and pour, where you basically give the lecturer back everything he gave you. there was no synthesizing. They didn't want to know what you thought about. They didn't care about your thoughts. So my first assessment, I did the exact thing. I crammed and I poured. And I got a C, maybe even less than a C. I got a really bad grade. And that for me was quite, why did I get a bad grade? So I went to the lecturer and I asked, what did I not do right here so that I can improve? And he said, well, you didn't really show any sophistication in your thinking. And that was the start for me. That was when I knew it's a different game now. And I very quickly adjusted. I think that's one thing I do admire about the Nigerian spirit. We are very, very adaptable, and we're very clever. I very quickly adapted, finished my master's. I ended up, that was the only C I ever got. Everything else was an A or a B. And I ended up graduating with a distinction. And then the lab where I did my master's dissertation, they had a space for a PhD candidate. It wasn't funded at the time. There was a promise to look for funds along the way. So my supervisor asked if I was interested in the position. That was in immunology. That was the very first time I had encountered immunology that way. The very first time I heard about CD, anything. The very first time as well that I held like a proper pipette. And I was quite flattered that after 3 months in her lab, she was willing to offer me a PhD position. So I took it. And again, I was fortunate that my family could afford the international fees at that time, with some difficulty because I had to work a number of jobs. In fact, there was a time when I had a tally of about 6 jobs, all of them 0 hours. And so I had to balance all my hours to make sure that I wasn't going above the stipulation for international students. So, but I did need the hours. I needed the 20 hours every week in order for me to be able to meet up with my living expenses. So it wasn't without any difficulties, but it was something I was determined to do. It was something that I wanted to do, and I was glad that I had the opportunity to do it, and I took it. So I did a PhD, and after my PhD, I applied for a job at the University of Oxford, and I got it. It was my very first application, but it was like that job was tailored for me. It was exactly what I had done. Yeah, so I spent 8 years at the University of Oxford as a postdoctoral scientist, moving from a postdoc to a senior postdoc, and eventually the lead immunologist on a clinical trial that's still going until today, which is quite exciting. And then COVID happened, and I did a lot of work with COVID at that time. And then decided it was time to think about my career again. My career, where did I want to go with my career? And then I had an opportunity in industry as a group leader, which was the natural next step for me. So I took that opportunity and I started a company called Adaptimmune as a group leader for cell profiling, and then moved to another company as a group leader for immunology, and then eventually came into my current company as an associate director for flow cytometry.
So maybe we should just talk more about what you've been doing so far in the field of immunology, starting from your PhD work, of course, and what was like the big question you were trying to address? What did you find out, and why would you think your findings would be important, or what contributions did those findings make to the field?
So for my PhD, the question I had was trying to understand how antigen-specific antibodies come about. So I imagine your audience are immunologists, but just in case they are not, an antigen is what the immune system sees. And all of your immune response would either start by being very non-specific, which is like the immediate responses you get in the first few hours that sort of gives you all the fever and the swelling and everything that you get, to be more and more specific, hence the swelling, hence the fever, because all of that is trying to recruit your specific arm of the immune response to that site of injury or insult, as we call it, immunological insult. So I was trying to understand how that specific arm of the immune response comes about and why in my mouse model it didn't happen. And so, like I was saying, we had sort of given them ova, which we use as a model antigen. Ova is ovalbumin, short form of ovalbumin. I think it's an egg protein. And they did not make ova-specific responses. So ova would make them, it would engage the immune system without causing any pathology. It doesn't, you don't get any sort of illness in the mice. And so we moved on to the next step by sort of really trying to challenge the immune system in those mice, and we did an infection with vaccinia virus. And then we found that with vaccinia virus, you know, the clinical outcome in our transgenic mice was much worse. So we were using the weight loss, we were tracking the weight loss across days. It was much worse in our transgenic mice than in our wild type mice. And of course, they didn't develop antibodies, which would be made by the B cells. They didn't develop T cell specific responses. So, you know, that sort of indicated to us that there was something connected with the adaptive immune response. And then we started to dig deeper. And like I said, we were doing a lot of histology then. So we would collect the tissues and do H&E staining and immunofluorescence staining. And at the time, I remember, these days people are doing immunofluorescence staining with like 10 colors or more. You know, it's brilliant. You see it and it's just beautiful. When I did it, 3 colors, and I was the star of the department because I had the 3-color immunofluorescence staining. So I did the H&E was the first. So we would first do H&E, which is just staining it with hematoxylin and eosine, and then looking at the pattern of the cells to sort of, every cell based on the proportion of the nucleus to the cytoplasm, they take a shape when you stain them. And so this is what pathologists would use to say, you've got this cell in here or you've got that cell in there. So I had a look, and I had to double as a pathologist as well at that time. And we could see a lot of T cells, and they are quite characteristic because they have a really big nucleus. But also what was interesting was that when we looked at our lymphoid tissues, so your lymphoid tissues would be your spleen, the lymph nodes, those would be all secondary lymphoid tissues. Tonsils are also secondary lymphoid tissues. Okay. Then you have the tertiary ones that grow at the site of infection. Those ones come up and go. Spleen is another one. Maybe I mentioned that already. So I was taking the spleen, I was taking the lymph nodes, and then doing H&E on them. And I found that the germinal center was completely atrophied as they get older and older. So the germinal center is quite organized. It's a really organized structure in the secondary lymphoid tissue, where you have your B cells in one region, your T cells in another region. Then you have a border where the T cells and B cells meet and then exchange information. And then the B cells then go away and make specific antibodies that they have been instructed by the T cells. And so, when I found that it was completely, the architecture was completely mashed, that sort of started to give us a clue as to what was happening. So I then followed that with immunofluorescence staining, and really you could see the T cells were not where they should be, the B cells were not where they should be. So how is that communication going to happen, right? So it made sense that we were not having our antigen-specific antibodies because that would be made by the communication between T follicular helper cells and the follicular B cells in the B cell zones. And so we then dug further and did some molecular work, and we found out that what was happening was that when we took out those genes, one of the instructors of the T follicular helper cell lineages did not come up. So that particular gene, it's a promoter, it didn't come up, or a transcription factor, sorry, it didn't come up. And that's because our gene works upstream of that gene. So our gene would go up or get upregulated. Then that then activates the promoter of BCL6, which is the transcription factor for TFH cells, T follicular helper cells. Those get activated and then move to the border of the T and B cells in the germinal, in the follicular region. and then have a crosstalk. We call it a cognate engagement with the B cells, and then tell the B cells, yes, I can confirm that this antigen you've seen is what I want you to make an antibody from. And then the B cells take that instruction and say, thank you very much, and go into the dark zone and start to churn out antibodies. They start to churn out these highly specific antibodies. So we didn't get that, and we found out the reason why we didn't get that. And then my postdoc at the University of Oxford was looking at this T follicular helper cells, but now in the context of humans and in the context of HIV. So I was working with this group of HIV infected people from Africa, and these samples were collected very early in the 90s, maybe some even in the 80s. So they're quite precious samples. These people did not go on the standard of care, the antiretroviral therapy, because that was not available at the time. Now the regulations are you have to put them on antiretroviral therapy as soon as the person is diagnosed. So that's the standard of care. For you not to put a HIV patient on antiretroviral therapy, you need very, very, very strong justification for that. And I don't think any ethical body is going to give you the approval for that. So those samples are considered really precious. But what was really interesting about the cohorts that I was looking at was that some of them developed these very broadly neutralizing antibodies, and some of them did not. And these very broadly neutralizing antibodies, it's known that they arise from the interaction between follicular helper T cells that I was working on and B cell interaction. So hence the reason why I came, because I was now, I knew a lot about this cell type thanks to my PhD. So my role there was to understand what was the differences in the people who made this broadly neutralizing antibodies and the people who didn't, with the idea being that we would reverse engineer that into a vaccine, a prophylactic vaccine, and then administer it into people who are high-risk populations, really. So, you can imagine what would be a high-risk population for HIV, maybe sex workers, or, or in some places it would also be men who have sex with men. So we will administer that into high-risk populations, and then hopefully they would develop this broadly neutralizing antibodies before they get infected, because this broadly neutralizing antibodies that develop in HIV-infected patients, it comes with a catch. By the time it develops, first of all, it takes a long time to develop, about 2-3 years into infection. It needs a high antigen load and also a high antigen diversity to develop. But also, at the time it comes up, it has no clinical benefits to them, because HIV mutates very quickly and their virus has already mutated from recognition by those antibodies. But if you can make it before, at the very start, before, viral escape, there's a chance that you might be able to target it. You might be able to target a virus and clear out infection. Even if it's not a sterilizing cure, at least you might get a functional cure. So that was what we were trying to do. And I worked on that for 5 years. It was a really difficult project because, like I said, these people had never been on antiretroviral therapy. We know that HIV affects CD4 cells. The cells that I was looking at, Tfh cells, they're about 5 to 20% of CD4 cells. So 5% if you're looking at a Caucasian cohort, and 20% if you're looking at an African cohort. So, and I was optimizing some of my assays in Caucasian cohort, so not very much to start with, but it was a really difficult project. And then after I did that for a few years, and I did a lot of assay development, I had to develop a lot of new assays to answer my question because of the challenge of that particular question. That equipped me with a lot that has served me for the rest of my career, really, because I learned a lot about flow cytometry there. I learned a lot about immunoassays there. I learned a lot about failing and getting up there, being resilient. I learned a lot about as someone put it then, being a catapult, where you have to pull yourself back sometimes to really push forward. there was, it was a very, very interesting period. And then I moved to another job, still on HIV, still in the university, and as a senior postdoc this time around. But what we were looking at was, instead of a prophylactic cure that would administer before people got infected, we're now looking at a therapeutic modality where what we wanted to do was, we had this cohort of HIV infected people who were already on therapy. We wanted to take bNAbs that have been made, so broadly neutralizing antibodies that have been made already, and engineered such that they last very long. And these were very broadly neutralizing antibodies because they could neutralize up to, I think, 98% of currently circulating strains of HIV, or had to neutralize strains of HIV. We get that, and then we infuse it into HIV infected subjects. And then we were very fortunate to be able to get ethical approval to then take these subjects off antiretroviral therapy. That was the strength. It was such a good study, such a good clinical trial. It is still running at the moment. So, it's so promising that we're able to get ethical approval to take people off the standard of care. And the idea is that, we're not trying to get sterilizing cure, because we know that that's almost impossible because of the latent reservoir that HIV forms. We were trying to get some kind of functional cure, where you can get people to take one infusion or two infusions a year, one infusion every 6 months, and they don't have to take a pill a day. And if you can imagine the utility of that, you think about remote areas where it's difficult to get the medicine to them, or it's difficult for them to come out to get the medicines. If you think about places where HIV is still stigmatized, where people don't want to know that, people don't want to associate with people who are infected. Mm-hmm. Yes, yeah, exactly. Don't want to associate with people who are infected. Giving them one infusion every 6 months is giving them a new lease of life. If you think about, just the quality of life of having to take a pill a day, that is difficult. That is very, very difficult. I mean, even for people who are ill for like a short time and they have to go on an antibiotics course for 7 days, sometimes you still miss some of your tablets, so to have to do it every day for the rest of your life, that's difficult. But also there's because of that problem with adherence, we have things like people not responding very well to antiretroviral therapy, because you're on it, off it, you're on it, off it, so you're giving the virus time to sort of develop a resistance to it, yeah, which we don't want. So there's a number of different public health reasons that sort of necessitated the sort of research that I was doing, and then COVID came, and everything had to stop, and we all had to pivot to COVID. And the great thing about the research during COVID was that we could all take our tools, from everything from the assays that we're doing in the labs to measure immune response, to the technologies that we had developed for HIV that we were either trying to get there, or we had got there and we had done clinical trials and it failed. We were able to take all of that and throw it at COVID. And this is the reason why the COVID vaccine came out so quickly, because we had all the technology. We had tried it. We had either tried it or we're in the process of trying it for HIV. The mRNA vaccines were scheduled to be tried for HIV in some other studies years ago, so we were on our way there. And so when COVID came, we were able to take the opportunities as a community of scientists all over the world. We were able to take the information we had learned, the lessons we had learned from HIV, and throw all of them into COVID, and all hands were on deck. Nobody, I assure you, nobody was doing anything else. Everybody was working on COVID. So that meant that the pace was really, really fast. So I did a lot of the COVID work. I built a team of scientists, and we did a lot of the characterization of the COVID responses at the start, because we wanted to understand, right? This was like the HIV of the 80s. We wanted to understand what was going on. So we first started to characterize what was happening in the immune system, what were the immunogenic epitopes in COVID, do people have cross-reactive responses to SARS-CoV-2, how did the different sort of clinical symptoms impact how people responded immunologically, and also how did that impact their lives after COVID? How did that were there sex differences in COVID? We were asking all those questions at once. So it was a very, very busy time. And then after COVID, I think I just was completely burnt out of academia. I felt like I had hit some kind of glass ceiling, the proverbial glass ceiling. I was in this elitist institution. I mean, it's great, it's brilliant, it's you well funded, but the opportunities are quite, they're not there aren't very many opportunities. So I wanted to take myself to a place where the opportunities were a bit broader for me, and industry came calling, and so I moved to industry to lead a group of at the time 3 scientists in profiling immune responses in cancer immunotherapy, and that was really exciting. Suddenly I wasn't living the life of an academic anymore. I wasn't I had a bit more change in my pocket after my monthly salary, which was good. I had more time to myself. It wasn't crazy. You have to read this paper. You haven't read this paper. It wasn't publish and perish anymore, or it wasn't a moving goalpost anymore. It was clear, the career part, but it was also a steep learning curve moving from an academic environment to a biotech environment or industry. And then a few months after I joined, the entire biotech industry was going through some really difficult time financially as a result of COVID and a number of other things. So my company was restructuring. And so I decided, I'm going to jump the ship because I thought I was going to lose my position. So I started to look for another job. But I was fortunate that I didn't lose my position after the restructure in the company I was with, but also I had another job. So I then had 2 options, which again, I was very grateful for, because there were people without jobs at that time. But I chose the other one because it was exciting. It was in gene therapy. I chose the new job. It was in gene therapy. It was very exciting for me. They had like really interesting technology, a really interesting platform. I wanted to understand gene therapy a bit more. I wanted to be in that field. I thought I had spent quite a long time in the sort of immunotherapy field. So I wanted to veer a bit out. And I was going to be doing immunology. So I was in my comfort zone, but learning a lot about gene therapy. And so I joined that company, and then 2.5 months into joining the company, they had a restructure as well, and my role was completely cut off. So I had well and truly lost my job this time around. And so we were given 2.5 months to sort of clear things off and wrap things up and leave. So I ended up staying in that company for about 5.5 months. And then I started to look for another job, as you would. And I was quite fortunate that I then got this really perfect opportunity to join an amazing team, that's trying to build a lab from the ground up. I had built a team before, I had semi-built a lab before, but now I was, doing it completely from the ground up. So that was great. Thank you.
For someone who's already working in Moderna, you've been in academia, you've been in industry. I believe there are listeners who are wondering, how do I get out of academia, or how do I navigate this transition from academia to industry? So you can just share a few tips for someone who might be asking that kind of, who want to find out how to, make that move.
Okay. I'll say that making the move from academia to industry is not always easy. It's a completely different mindset. In academia, you're focused on, academic research is very much, there's no end to it. You just keep going and going and going and going, and it's very pedantic. It's very, what are the words that I'm looking for? I don't, without saying academic, it's very academic. It just keeps going. While industry research it's got a focus. The business is always your primary focus. The end goal, the patients are another focus, because you're much closer to the clinics. You're trying to make sure that you can get your company to the point where you're able to be useful to the population that your therapies are targeting. So it's a completely different mindset. Sometimes the academic mindset, you get stuck, that you don't know when to let go. Industry mindset, there's a go no go decision that needs to be made. Does this look feasible? If it doesn't, okay, it's time to leave it now. It's time to focus on something else. A lot of people in academia think when they move to industry, they lose their altruism. They've gone to the dark side, they've gone to make a bit more money, when there's absolutely nothing wrong with making money. But you're also you still retain that usefulness to society, because you're still doing things that are important, whether you're at the start of the drug development process in R&D, where you're looking at the technology, you're optimizing the platform, or you're looking, you're further along in the sort of middle bit where you're doing preclinical testing using either in vitro models like cells, or in vivo models like mice, or non-human primates, or you're further along even, and you're in the clinical side of things, where now you're testing your therapies after they've been to a clinical trial in to check for safety and efficacy of your product. There's so much that you're doing that's contributing to the benefit of patients on the other end. There's a lot more, I wouldn't quantify it with more or less, but there's a lot of good that you're still doing. If you want to move from academia to industry, I would say that you need to keep an open mind. You need to be ready to embrace the industry life. You need to let go of the notion that publication is how you determine your worth, because you might not be in an area where you will publish as often. And the bane of academic life is, oh, I've just got this paper published today in Nature, or in Science, or in Cell. It could happen in industry, but you need to reframe your thinking. You need to change your mindset. And I personally, I've always loved to do work where I can see the benefit. I can see how it would benefit people. Basic research is lovely. Everything we do starts from basic research somewhere. But I have always loved the sort of translational space, and I have done basic research. My first 9 years of research was basic research, but I've always preferred the translational phase. I've always been interested in the diseases, interested in the people side of things, the patient side of things, so I get that in industry. And if that's what you like, you'd get that in pretty much any company you join, because every company has got a target patient population that they are looking into.
So like you mentioned earlier on, you have this interest in engaging the public, in supporting young and emerging scientists, and of course you have that keen interest. So I want to ask, for someone like you, started out in Nigeria, in Africa, and how do you still manage to stay in touch, to just stay in touch with happenings in the country, happenings in the continent, connect with other scientists to ensure that you are also supporting what's going on over here. And I think we can also learn from your experience doing that, and see how other scientists in the diaspora can also contribute to supporting research, supporting education over here in Nigeria.
The short answer is that I haven't. I haven't done that.
Okay.
So I have done a lot of sort of support here, a lot of public engagement here, a lot of mentoring here. And when I've had Nigerian students or African students that have found themselves in the environment that I have been, I have to say that I haven't been focused on just Nigerian students. I have been focused on people that I know that are from the continent, because I think, you know, I can, my experiences are can cut across the entire continent. So I think there's a lot of similarities with people from the African continent. I try to support them here, whether it's by helping them navigate the space, or signposting them to where they can get help, or just by tutoring them through stuff. I haven't done a lot with Nigeria, and that's not because of it's not for a lack of wanting to do, or it really has been at the start, I think, not knowing how to do it. So I had, and by not knowing how to do it, some of it was work that I needed to do to understand, and some of it was also could also be made better from the Nigerian side. So I started in maybe 2015 or something like that, with trying to have a mentorship program where I was getting a lot of scientists in the UK, in the US, and in Europe that were of Nigerian origin, and I was trying to link them up with students in Nigeria. And that came apart because, you know, people say yes, they want to do stuff, but people don't often have the time to dedicate to do stuff. And I would confess and say I didn't have the time either to follow people through like that, because I also had a full-time job. And so that fell apart. I remember we had a few meetings, we had like a charter, we had things we wanted to do. And then on my part, I did think of linking up with my university, Ahmadu Bello University in Zaria, and doing a guest lecture there, you know, a few times a year, or just opening myself up to say, if you've got any questions about XYZ, you can come ask me. But I thought about, and this was, don't forget that, you know, there's been a bust in the use of the internet now, but certainly at that time, the possibility of doing lectures virtually was non-existent. This is 2015. A lot of things became virtual during and after COVID, you know, so the possibility of doing lectures virtually was non-existent. And then there was all the crisis going on in the north with Boko Haram. And I said, there's no way I'm going to put myself in that situation. You know, I don't want to go back and then put myself in trouble by, you know, going to deliver a lecture there. But also there was the funding. Do I fund it on my own, or do I seek for funds? Seeking for fund is quite a task. It is time consuming. And I didn't really have the time to invest to figure out where I can seek for this task funds. I did email some prominent Nigerians at the time. Admittedly, they were in the entertainment industry, because those were the ones that I thought I could reach out to with some of these ideas. I didn't get any reply back. And so, you know, I had no funding, I had no sponsorship, I had a full-time job that I needed to pay attention to. I was very early in my career. I had this dream, I had no way to fulfill it. So I thought, okay, maybe you wouldn't work because it's in the north. How about Unilag? Lagos is pretty safe. I lived in Lagos. By safe now, you know, it's safe from Boko Haram at least. And I can navigate Lagos. I know how to navigate Lagos. So yeah, maybe I could do Lagos. But I didn't have any contacts in Lagos. I didn't go to the University of Lagos. I didn't know anybody there. So it didn't go further. So I'm not pleased about it, but it just never went further. And it's something that always, I still think about. And hence the reason why, as soon as I got your email, I thought, yeah, I'm not going to have the time, but this is something that I wanted. And so one of the things that I thought we really lacked was that knowledge of where the field was, knowledge of what science is. Maybe I speak for myself, but I could have done a lot with a bit more um, um, liberty in my learning, a bit less authoritarian approach from my lecturers, where I had to give them what they had given me in the class. I could have done with a bit more, with being challenged a bit more, being pushed a bit more, rather than, just today I don't remember very much about what I did in my bachelor's. That's because it was short-term memory. I memorized it. I put it on the exam paper. I got the results I needed to get, and I was out. But the things where I've sat down and I've had to think, I've had to synthesize and come up with my own opinions, come up with my own philosophy on things, those things have stayed. So I think I could have done with that type of learning. One of the things that I thought of establishing in the mentoring club was a journal club, where, because we were in academic institutions, we had access to journals, and I didn't quite know what the copyright laws would be then, and I'm not sure that I even know it now, but because it never went that far, I didn't investigate, but I was hoping I could share it, especially open access journals. I could share that with students in Nigeria, and then we could start first by, how do you read a paper to understand? How, and then we could go into, what has this paper told us? And then from there, you can go into what would be the natural next step with this sort of work, but I didn't have the critical mass to establish it. I didn't know my way around anything. And, I'm a bit of a nerd. So I didn't have a lot of friends to say, oh, I've got this person or that person that I can reach out to. No, I just, yeah. So that killed the project right there. But that's still something that I think of all the time. And I keep thinking, when I have a bit more time, when I have a bit more time. But as it turns out, we're never going to have the time we want. So, maybe it's time to just bite the bullet and start something.
Yeah, I agree. So, but I'm going to disagree with your initial response. You said you've not done anything. Well, I think you've made some really interesting and commendable steps.
I could have been a bit more persistent. I could have been a bit more persistent. But...
yeah, but maybe you can still try again now, like you just mentioned.
Yeah.
While you are closing up the response. So thank you so much. And I believe even other individuals who might want to try out something like this, this could be the motivation they need. Okay, someone else has thought of this kind of idea, so maybe I can still try something similar. So yeah, thanks for sharing.
Yeah, and if there are people that want to do it, I'm happy to be reached out to. Mm-hmm. You can reach out to me and we can try to flesh out something. If there's a critical mass of students somewhere that want something like that, feel free to reach out, and I can attend the journal clubs and I can help with the interpretation of some of the papers, and I can provide that sort of someone who's been through it view, give you, that's, let's, dissect the science here. We can talk about technology. I'm very, very passionate about the learning of the next generation. I'm very passionate about, how science is instructed, how people are learning science, because you want to know that the next generation will be okay. The classic phrase, the kids are all right. You really want to know that the kids are all right. Yeah. So, I'm happy to work on it. If anyone's interested, they can reach out.
Yeah, sure. But I just wanted you to just quickly highlight some of the challenges you've had to grapple with, and maybe what has worked for you in handling them. So maybe, do you try to climb over the walls, or do you walk through them? Do you tear them down? So what are kind of some of the techniques or approaches you've used to address some of those challenges that especially the other women in your setting or even young girls from Africa or Nigeria can learn from?
Okay, that's a really good question, and I don't know that I'm gonna do it justice. So like I say, I do appreciate my privilege that first got me here. It's very, very disheartening for me that there are people, there's potential, there's talent, there are people who want to study abroad, you know, who are way more intelligent than anybody I have ever met, you know, people with maybe the cure to cancer or certain types of cancer or even HIV in their head, just locked up in there as a potential and can't get it. So you know, I'm very grateful for the opportunities that I have had. They didn't come without their challenges. Some of them I have mentioned here, like the fact that, you know, the learnings, I didn't think my education was sufficient for me. There was also the financial challenge that even though I could pay my tuition, I had to leave. I had to have my living expenses. And there was a time in my third year of my PhD, you know, I had done first year, second year. Third year, I was almost withdrawn from the university because I couldn't afford my fees. And I know that at the time, you know, um, a lot of my savings, a lot of my savings from, you know, family and it all had to come together to pull me through the edge at that time. So, you know, there was that challenge. There's working and schooling. If you can avoid it, you know, you can't have two masters. You cannot have two masters. And I know that a lot of Nigerians, because of the situations that they cannot help, they plan, you know, when I come here, I'm gonna work and then I'm gonna get my degree. If you can do that, fantastic. But I would say now that it's a very, very challenging situation, you know, because you're trying to work. I think the regulations are still 20 hours a week. You think 20 hours a week is not a lot, but it's a lot when you're working because, you know, you are really working here. There's no skiving off work or coming to work and sign your name and you go away. There's nothing like that. You are really working. Sometimes you're on your feet. I remember I was an overnight staff at the library because I didn't want to give up my day. So I was giving up my weekends and I was giving up my sleep. So I would work as an overnight staff in the library, sometimes after doing an entire Saturday as a tour guide. And then, you know, the tour guide would be 8 hours. Then the overnight shift was 12 hours. That made up my 20 hours for the week. And it would be, you know, the tour was on Saturday. The overnight shift was on Sunday to a Monday. And you're completely like a zombie for the next 2 days because you haven't slept. And then, you know, and I'll be in the lab trying to still get my work done. So it's really tough. But what kept me going, and of course there's the cultural differences, there's the weather, there's the loneliness, you know. The Nigerian society is so vibrant, everybody is in your space, and you know, when you're in Nigeria it's a bit too much, but when you come out of here, you're so used to come out of there, sorry, you're so used to it, you're like, where's everybody? I want people in my space. Not so much for me because I don't like it so much, but
Yeah.
but you know, there's that loneliness to grapple with. There's the cultural differences to grapple with. So you know, lots of challenges, financial, psychological, cultural, lots of it. But what really pulled me through the first thing was I wanted it. I wanted it. You know, and this might be sad, but I thought there's nothing back at home for me. There's no, what am I going to do back in, you know, this is where the science is at. If I wanted to do science, I can't do that there, you know, and I'm a science girl at heart through and through. There was, you know, there was no question about what I wanted to do. I can't do that there. I want this. And there were days when, you know, I always say, just there are some days that you can see quite far into the future, and there are some days that you can't see beyond your next step. And that's enough. That is enough. I don't, I'm not trying to kill myself in the process. I take each day as it comes. The days when all I need to do is put one step in front of the other, that is what I'm going to do. The days when survival is just enough, that's exactly what I'm going to do. And then there is always, 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 I tell people this all the time, there would always come a period of thriving. You would always thrive eventually if you can, just persevere. Because you're thriving doesn't mean you're always going to thrive. It's like mountains and troughs. The good times would come, the bad times would come, then good times, then bad times. It would always happen. So it's just, what exactly do you want? Persevere. And one thing as well that I think the Nigerian spirit gives a lot of us is resilience. And sometimes it frustrates me because things are happening. The country is burning down, and they're like, last, we could be all right, or something like that. And you're like, no, maybe it's time to hold someone accountable. And they're like, not cruise, or and it frustrates me a lot. But that is the resilience in the Nigerian spirit. It's almost like all you need is a sleep as a Nigerian, and you reset, and you're like, we go out again. We do it again.
Yeah.
And then we have all these sayings that it makes me, I roll my eyes sometimes, but I have seen them come to play for me, come to work for me in a number of ways, where you're like, as long as you're not dead, you carry on. So I think the things for me, as a woman, as a female, as a Nigerian, as an African, it's know what you want, focus on what you want, keep putting one step in front of another. Consistency, persistence. I think a lot of people have said it, and maybe I'm preaching to the choir here, but it does work. And also to acknowledge that it's not going to be an easy journey. Some days would be good, some days would be terrible. On the days that are terrible, if you do not feel like going out of bed, by all means, stay in bed and wait for the days when things become better. But while you're in bed, do not give up. Do not give up. Take the rest you need. That's basically what I'm saying. If you need to cry on some days, cry your heart out. If you need to take rest on some days, take rest on some days, but do not stop. Just keep going, keep going.
Keep going.
And
okay.
Yes, and things will always change. It's a promise. Sometimes it looks like it would never. Sometimes it looks like you're in a hopeless situation. I promise you, things would always change.
Thank you. Thank you so much. We'll get your closing remarks for the episode.
I guess for me, it's know what you want. Don't be afraid to challenge the status quo. Don't be afraid to think outside of yourself. And by that, I mean, sometimes, as people, we think, oh, I'm this, I'm that, I'm that, I'm not good enough. I'm not, whatever enough that we, don't be afraid to dream of yourself as something massive. Just dream. They say aim for the stars, and if you fail to get to the stars, at least you get to the clouds or something like that, right? Don't be afraid to do that. My absolute favorite poem is a poem by this lady called Marianne Williamson's. Yeah, I think so.
Yeah.
And it's called Our Deepest Fear. And there's a part of it that says, how our deepest fear is that we're inadequate and that we are, all the things we tell ourselves. Yeah. But really, that is it. You are powerful if you do really go into who you are and explore your potential. There's almost nothing that can stop you. You might not get it, but at least try. And sometimes, I tell you what, sometimes what opens the door is the confidence for you to even, because people are like, how dare you? There must be something about you that made you knock on this door, because how dare you? And at those times, just hold your head up high. You might be pissing yourself to, with fear, but just hold your head up high and maintain your space. So take your space, maintain your space on the table. If you've got one leg in, use that leg in to push your entire body in.
Yeah.
Take your space. But I would always say, while you take your space, also make space for other people. So be humble enough to know that you're not the only one in there. So take your space, but make space.
For others.
For others, yeah, absolutely. And if you find yourself in as a scientist in any scientific endeavor, if you have the opportunity, learn a technique. Become a specialist in a technique. For me, that technique was flow cytometry and immunoassays. I have left flow cytometry in some of my jobs and focused on other things. But what I can always fall back on is flow cytometry. And I've gone, in and out of it, but it's always going to be a tangible skill set for me, a marketable skill set for me. So always learn a technology, whether it's mass spectrometry, flow cytometry, or immunoassay techniques. I would, learn something that requires the use of a specialist machinery, because it always makes you marketable. If it's the transcriptomics, if it's coding, if you want to go into the bioinformatics, learn something that requires a specialist piece of machinery, and you'll always be marketable.
Thank you for listening to this episode on Immuno Africa. If you enjoyed it, please feel free to share it with your network. You will learn first about future episodes and get other immunology related updates by following Immuno Africa _ other Immunology in Africa podcast on social media. See you on the next episode. Bye.